Now, let's see the other type of tissue, that is nervous tissue. Nervous tissue consists of two categories of cells, which can be called as excitable cells and non-excitable cells. Under excitable cells, we will be studying about neurons. Under non-excitable cells, we will be studying about neuroglial cells, which also form the supporting tissue of excitable neurons or nervous system. Now, let's see about excitable cells or neurons. Neurons are the functional units of nervous system, and these are called as excitable cells because they can generate an impulse, or they can initiate an impulse, or they can receive an impulse, they can conduct an impulse, and they can transmit an impulse. Hence, these are called as excitable cells, which carry information from one nerve to the other nerve and from the nerves to the effector organ. And these neurons consist of cell body and its processes. Its processes involves one axon and many dendrites. In common, these neurons are also called as nerve cells, and these nerve cells, they do not multiply, but they only can grow, and they also can synthesize their own energy, ATP, from the glucose. Nerve impulses can be initiated from any stimulus, and this stimulus can be an external stimulus or an internal stimulus. For example, light and touch are the external stimulus, whereas change in rate of respiration with the change in carbon dioxide concentrations in the blood acts as an internal stimulus. And this transmission of impulses along the nerves and between the nerves can be electrical or chemical. Electrical transport involves changes in membrane potential across the nerve cell membrane, whereas chemical transport or transmission involves exchange of neurotransmitters from one neuron to the other neuron. So, a transmission of impulse within a nerve is always electrical, whereas a transmission of an impulse from one nerve to the other nerve is always through chemical transmission. Now let's see about the structure of nerve cells. We have seen nerve cell consists of cell body and various processes such as one axon and many dendrites. Cell body varies in shape between different neurons, and these constitute the grey matter. Groups of cell bodies are present in the peripheral regions in the brain, which makes the grey matter to be appearing in the peripheral regions, whereas groups of cell bodies are present in the middle of the spinal cord, which makes the grey matter in the middle. And groups of cell bodies in the central nervous system are called as nuclei, and groups of cell bodies in the peripheral nervous system are called as ganglia. Now let's see about axon. Each neuron has one axon, and it originates from the cell body at the axon hillock. The membrane of the axon is called as axolemma, and based on the presence of myelin sheath, these neurons are categorized into myelinated neurons and non-myelinated neurons, which can be categorized based on the presence of myelin sheath on the axon. Myelinated neurons are generally long. Large axons and nerves of peripheral neurons consists of myelin sheath. Generally, there are a series of Schwann cells which wrap around the axolemma, and under the Schwann cell membrane consists of a pigment called as myelin. And there are some regions in the axon which are not covered by this myelin sheath and where the axolemma is exposed. And this region, it is called as nodes of Ranvier. Impulse transmission across such sort of myelinated neurons will be fast, and this is called as saltatory type of impulse conduction. Whereas small fibers and axons of postganglionic neurons, they do not consist of this Schwann shells, and hence they are called as non-myelinated neurons. And transmission of impulse along these non-myelinated neurons is generally slow, and it is called as continuous propagation. Whereas many axons of non-myelinated neurons are enclosed in one Schwann cell. The dendrites are many in number, and these are highly branched, and these dendrites act as synapses in the case of transmission of impulses as that of motor impulses, whereas in the case of receiving impulses as in the sensory nerves, they act as receptors.
In this video, we will talk about the primary cell of the nervous system, the neuron. We will talk about the structure of neurons and also how they come together to form an entire network of nerve cells. The neuron is a very fascinating and vital portion of the nervous system because it allows the nervous system to communicate with the rest of the body. The structure of the neuron offers key features to help it transmit messages across relatively long distances. To begin, the neuron has a cell body, which is also known as the soma, and which houses the cell nucleus. Next are the dendrites, which are tree-like branches that extend off of the soma. These are used by the neuron to detect both chemical and electrical stimuli. In the center of the neuron is the axon. The axon is the longest extension from the soma and can be up to 1 meter in length. It serves as the primary communication pipeline from one end of the neuron to the other. The axon hillock is the junction that connects the axon to the cell body. Covering the axon are myelin sheaths. Myelin sheaths are sections of fatty tissue that help to protect the axon and also help to speed up signal conduction along the axon. The gaps between myelin sheaths are called the nodes of Ranvier. These also aid in signal conduction, and they also allow nutrients and waste to enter and leave the axon. Finally, at the end of the axon are extensions that branch off to form several axon terminals.
Next we'll see neuroglial cells. Neuroglial cells are also called as non-excitable cells because unlike neurons, these do not transmit any impulse or they cannot generate an impulse. Hence, these are called as non-excitable cells. And these are generally the supporting tissues of nervous system. There are 4 different types of neuroglial cells. They are astrocytes, oligodendrocytes, ependymal cells, and microglia. Let's see them in detail.
So glial cells are the supportive cells which support neurons in their function of communication within the nervous system. The word glial actually comes from the Greek meaning glue. So when they were first discovered, it was thought that the glial cells actually acted as a connective substance within the nervous system, holding it all together. Glial cells are not directly involved in information processing, but they are essential in providing support to the neurons. So there's quite a few different types of glial cells. There are 6 types, and you've got 4 in the central nervous system and 2 in the peripheral nervous system. So in the central nervous system, you've got the astrocyte, you've got the oligodendrocyte, the microglial cell, and you've got the ependymal cell. In the peripheral nervous system, you've got 2 types of glial cell. You've got the satellite cell, and you've also got the Schwann cell.
Astrocytes, these are the main supporting tissue of nervous system, and these consists of star-shaped cells with fine branches with foot processes at their ends. And generally, astrocytes are present in large number in the central nervous system, especially around the blood vessels which are supplied to nervous tissue. The foot processes of astrocytes, they form a layer around the blood capillaries so that the blood which is supplied to the nervous tissue does not have direct contact with that of nervous tissue. And this endothelium of blood vessels is generally tightly associated with each other. And these endothelial cells of the blood vessels which are supplied to the nervous tissue are closely packed with each other, and the foot processes forming a sheath together form the blood-brain barrier, and they restrict the movement of substances from the blood into the nervous tissue. This blood-brain barrier is selective to substances such as blood glucose, oxygen, and lipid-soluble substances, whereas large molecules, inorganic ions, cannot be transported between the blood and nervous tissue.
So first of all, we'll take a look at the astrocyte. So with the astrocyte, immediately striking is the shape of it. It's got this central body and it's got these projections out from it. So it's kind of got this star shape. So the word astrocyte comes from the Greek, unsurprisingly. Like everything in anatomy is Latin or Greek. So astro refers to astron, the Greek word for star. So it's got these extensions from the cell body. And these aren't axons and they're not dendrites, they're simply extensions. So the function of the astrocyte is purely supportive. What the astrocyte does is it helps to maintain chemical concentrations in the extracellular space. It also is involved in removing waste, and it has some reparative function, so it can react to tissue damage. And another crucial function of the astrocyte is its contribution to the blood-brain barrier. So the brain is a protected organ. Substances which are circulating within the circulatory system, within the blood, can't just enter the central nervous system at will. In fact, a lot of substances have difficulty passing from the blood into the brain. So this barrier separating the blood from the brain is the blood-brain barrier. So it's not shown on this diagram, but the astrocytes actually have these vascular foot processes which can wrap around capillaries and provide this blood-brain barrier. So I've just drawn on an illustration of a cross-section of a capillary, and if you just imagine the extension of this foot process wrapping around the capillary like this, and you might also have the foot process of another astrocyte wrapping around to form a complete barrier. So similar to the astrocytes are the satellite cells in the peripheral nervous system. So you've got astrocytes in the central nervous system, and now we're talking about satellite cells in the peripheral nervous system. And the reason I've grouped these two together is that they have a similar function. So they have a broad supportive role. And satellite cells are found surrounding sensory and autonomic ganglia in the peripheral nervous system. So autonomic refers to the parasympathetic and sympathetic divisions of the nervous system. So these cells are thought to regulate the microenvironment of those ganglia in the peripheral nervous system.
The next cells are oligodendrocytes. Oligodendrocytes are smaller than astrocytes, and these are abundantly found around the nervous tissue and also along the myelinated neurons. Oligodendrocytes are involved in maintenance of myelin sheath similar to that of Schwann cells, which are involved in maintenance of myelin in peripheral nerves.
Oligodendrocytes on the right, and you've got the Schwann cells on the left. So the oligodendrocytes on the right are found in the central nervous system, whereas the Schwann cells, which you can see on the left, are found in the peripheral nervous system. So the reason I've grouped these two together is because they share the same function of insulating axons. So they provide the myelin sheath around the axons. So I've just outlined the cell body of the oligodendrocyte in red. And you can see how it extends from the cell body to actually encapsulate multiple axon segments. So if you contrast this to the Schwann cell on the left, the Schwann cell is a single cell that only encapsulates one single segment. So what is myelin? Well, it's this fatty lipid-rich substance. And the fat in the myelin is made from the phospholipids of the cell membrane of these two cell types, so the Schwann cells and the oligodendrocytes. And the purpose of myelin is to insulate the axon and prevent the electrical signal, the action potential, from escaping from the neuron. So as well as providing this insulating property, it also increases the speed of electrical impulse down the neuron. So I've just drawn a cross-section of an axon here, and I'm just going to illustrate on the different sort of layers of the Schwann cells. So close to the axon, it's layered very tightly together, and these tight layers don't contain cytoplasm. It's only this outer layer of the Schwann cell which contains the cytoplasm and the nucleus. So this outer layer of cytoplasm in the Schwann cell is called the neurolemma.
Next, we'll see ependymal cells. Ependymal cells are the cells which line the ventricles of the brain. Brain has 4 different ventricles, and these ventricles are lined with this ependymal cells, and they also line the central canal of spinal cord. These ependymal cells, they secrete cerebrospinal fluid, especially the choroid plexus, which is formed by these ependymal cells, secretes cerebrospinal fluid into the ventricles.
Ependymal cells. So these are similar in appearance to epithelial cells, and you can see how they've got this close relationship to one another. So the cells are tightly adherent to each other, just like epithelial cells. The function of ependymal cells is to filter blood to make cerebrospinal fluid, which is this fluid that flows through the CNS, and it lubricates neural structures. It acts as a protective kind of cushion for the neural structures. So cerebrospinal fluid, or CSF, is found to be produced in the brain ventricles. So ependymal cells are found in a specialized area within the brain ventricles known as the choroid plexus, which is where these cells are in contact with the blood vessels and they can filter the blood to create this CSF. So you'll notice also that there's these extensions from the ependymal cells, and these are known as cilia. So these cilia are structures which can waft the CSF through the brain ventricles and get it moving through the ventricular system.
The last type of neuroglial tissue is microglia. Microglia are small but numerous in number, and these microglia are similar to that of monocytes in blood. Monocytes migrate to the central nervous system before birth, where they are modified into microglia, and they have the same phagocytic function, and they are involved in phagocytosis of any foreign material which has entered into the nervous tissue, or removal of cell debris and microbes.
So on the left hand side of the screen we've got the microglial cell. So the name indicates something about it. Micro meaning small, so it's smaller than the other glial cells. And the function of the microglial cell is really similar to that of a macrophage. So it's to ingest cells and pathogens. So it's thought that the microglial cell may have originated as white blood cells, as they're very similar to the macrophages of the immune system. So microglial cells really have a role in immune support. So that's an overview of the different glial cells that can be found in the nervous system, and this table is really just to highlight what we've talked about. So you've got 6 glial cells, you've got 4 found in the central nervous system, 2 found in the peripheral nervous system.
This is regarding nervous tissue.